what animal models can be used to tell us is what's going wrong, why, where, and when does it go wrong? Because you have to think about all of those things in terms of thinking about therapies. So which cells, which organs, and we know in SDS already from the clinical picture that there's multiple organs affected in SDS. But we also need a little bit more detail than that. We need to know when do the concerns begin and what are they? What are the times that they respond? How are organs responding differently? And not only that, what are the patterns of those responses in terms of what actually happens in the organs to fail? Because we know from complex biology that organs can fail in different ways, even if you say there's a common underlying problem, which is certainly would be the SDS issue. We have a common underlying gene failure, but what happens? This is the variant that Esther had talked about that is an important common variant that's been seen in a lot of patients with SDS. The most models that exist now does not include the common splice variant. This common splice variant is a very interesting mutant because it results in a very small amount of normal protein. And I think, and it occurs in quite a few patients. So the best thing would be to help that work if that's feasible. And that we don't have with the mouse models we have right now.
This is, of course, a disease in which this gene is impaired in all cells of the body. So what we really need is not only the study of cells, but the study of animals, where all these cells come together and this mutation occurs in all the cells. And to begin modeling this, you need an animal model, and preferably a mouse model, because it's, as Johanna was saying, much closer to humans than, for example, a zebrafish model. We have made success, we have had success with the mouse models that exist so far in our lab. I think Johanna has really been a pioneer and a collaborator, but at the same time, there is a lot of room for improvement, mimicking mutations that are even more common in patients, generating mice that mimic more of the phenotypes of patients, and having animal models where we can actually test drugs. So I was very excited about this effort to try and build more and even better mouse models, because I do think that they may be really a key asset to improving our understanding of the disease, testing drugs, and then ultimately the outcome for our patients.
We've used Drosophila, so we've used a fly model, which is an SDS hypomorph, and we're using that to try to develop a drug screening program. But obviously, these are flies, and we absolutely need to move forwards into testing any compounds that come out of this study. We need to test them in mammalian cells and in a hypomorphic SPDS deficient mouse model. So again, we're really excited about the possibility of having this mouse to actually test, assuming we get some molecules at some point, to testing them in the context of the SPDS deficient mouse. Can we identify small molecules that can recapitulate this N106S mutation? So small molecules that would bind to EIF6 and promote its release from the ribosome in the presence of in SPDS deficient cells. So this is the strategy that we're actually pursuing at the moment, and so far the results look encouraging in that we've identified some small molecules that do indeed rescue, at least in partially, the phenotype of these SPS deficient flies. But it's absolutely essential to moving forwards to try to test this in a mammalian system, and so we're really looking forward to seeing these SPS deficient mice.
And so the goal again, is to create, to leverage the infrastructure at the Jackson Laboratory which I'll tell you about in just a second, to create this seamless pipeline to support the preclinical needs of the rare disease community. And we do that by making mouse models, characterizing mouse models, and doing the drug efficacy testing in those mouse models, where we can. So I'll say that just an introduction to our capabilities at JAX. We do have a lot of capabilities around accessibility of mouse models. We have one of the largest repositories in the world that contain, I think it's actually now over 12,000 mouse models, and many of which are live on the shelf. And we specifically make sure that we try very hard, especially in rare diseases, that even though there's only a handful of researchers around the world working with these mouse models, that we do make them available. And for mouse models that we generate in-house, we try very hard and are actually quite successful at not putting any licensing restrictions on these particular mouse models, so that the barrier for use in biopharma and biotech could be readily accessible. The last thing we want to do is put a big price tag between the people who are going to help create the therapeutics and then the ability to test those therapeutics. So these mouse models are made available widely to anybody in the world who wants to use them, and all you need to do is call and order them from our catalog. But in addition to the mouse models that we have in our catalogs, we have the ability to generate these models through our model generation service, which is quite extensive. We have a really large core for CRISPR-Cas9 and other technologies in which we generate these models. And then we have phenotyping capabilities. Our phenotyping capabilities around mice are pretty extensive. And we have the ability to not only make the models, but then look at the translationally relevant phenotypes that we see or the translationally relevant clinical presentations that we see in the kids, and see if we can then solicit and elicit those in the mouse models that we make, much like Johanna had told us, earlier in the day. And then I think, probably of interest is our in vivo pharmacology testing service. We do a lot of drug testing on mice. I cut my teeth on SMA for almost a decade, so I'm very familiar with how the preclinical and the pre-IND solution for those things involving preclinical pharmacology work. And so we offer not only the drug testing, but the safety and tox. And we have multiple grants from NIH, one of which we're working on called the JAX Center for Precision Genetics. And this is the mechanism by which we were able to fund Esther's request in order to make this splice mutation that we hope to have relatively soon. And just, briefly looking at the organization of the intron exon structure between mouse and human, there are differences, in this area where we want to make this splice mutation. So one of our strategies then is to humanize that area around exon 2 so that we can make a mouse model that has the exact same splicing mutations, as we see in the human population. And we're hoping that we get to work with all of you here on the call to help have this mouse model be impactful for therapeutic development in this field.
Yeah, like for example, being able to say the mouse exists would be a big asset on a grant application, because the funder will say, oh, this mouse exists, they can do this experiment. And that's a really important asset to have.
Yeah, I was going to say the other advantage is that there's a therapeutic strategy associated with the splicing mutations in terms of antisense oligonucleotide therapeutics, that can also be used in addition to, some strategies around gene therapy. So, if we can get that model, and as Johanna, you've mentioned, if we can get the phenotypes, that we need in them, we can start testing those specific therapeutic strategies in the mouse models as well. I think that's one of the things I'm most excited about in this regard. I've seen a lot of really successful rare diseases, really take off under those conditions. And I think we've got a great group of people that have a really deep knowledge base. And so getting the resources and the tools and the people together, and that's what we're doing here today.